All right, welcome back to the second part of the video lecture for chapter 2 here. Um, so at the end of the last video lecture, we had just finished talking about how we validate phylogenetic trees, and that's what this slide is that you see right here. The next thing to discuss, next major topic to discuss, is the concept of molecular clocks. So one thing that's interesting about, or that's helpful about phylogenetic trees, is that they show us the pattern of evolution, the branching pattern, for example, that you see here in this figure. Another useful aspect would be to have some idea of when those major branches or speciation events, as they're called, where one ancestral species splits into two descendant species. Another useful thing would be to know when that happened. In other words, can we date these speciation events, the splitting events that you see on these trees? And one way to do that is through the use of what's called a molecular clock. So absolute dates of divergence, exactly when something happened, with some margin of error, let's say, from a common ancestor can be determined directly by dating fossils, okay, a radiometric dating. In other words, using radioactive isotopes, for example, to figure out how long a fossil's been sitting in that rock. Those give us absolute dates, absolute rather than relative, right? Now, another way to do this is through the concept of a molecular clock. Let's say you're just dealing with DNA sequences and you're not dealing with fossils. Well, obviously then you can't use radiometric dating, so we need some other way to sort of estimate when these divergence events occur. And the molecular clock concept can be very helpful for that. So what is a molecular clock? Basically, it's the process of using known rates of mutation in certain genes to determine how much time must have passed since a descendant species diverged from its ancestor. All right. Let me say that again. The molecular clock is using known rates of mutation in certain genes to determine how much time must have passed since a descendant species diverged from its common ancestor. The idea is that if you know at what rate a gene mutates, and if that rate is known to be fairly constant, which it is for some genes, it's possible to back calculate how much time would be required to result in the differences in base sequences that we see for a given gene when compared between two species. In other words, it takes 1,000,000 years for a certain gene to develop a single mutation, and you see 3 mutational differences in that gene between two species, then you can estimate it's taken about 3,000,000 years. It's been about 3,000,000 years since they shared a common ancestor. That's the idea. Okay, so let's take a look here at an example of this. So for a molecular clock to work, mutation rates have to be relatively constant, okay? That's a key point. Also, there can be no homoplasy resulting from convergent or parallel evolution or reversal. For example, it's possible to have a DNA sequence where you have an A base, an adenine base that mutates to a cytosine base at one time, and then sometime later the cytosine mutates back into an adenine. That's a reversal, and that reversal hides that initial mutation. It's like it never occurred. It erases it. So that kind of thing can disrupt our estimates or make them less accurate. So we have to sort of be aware of that sort of thing and try and estimate when that happens. Okay. All right. So if we know how fast a gene mutates, how fast the clock ticks, in other words, we can estimate absolute times of divergence. All right, so look at this phylogeny over here on the right. Okay, so we have these 4 different extant species, currently living species, A, B, C, and D. And this phylogeny suggests that species D branched off the common ancestor earlier than C, B, or A did, right? It also suggests that A and B share a more recent common ancestor than either one does with species C or species D. So these two are more closely related. When you compare all 4 of them, C, B, and A are more closely related to each other than D is. D is more distantly related. All right. Also notice our hash marks here, horizontal hash marks. These indicate mutations. So if we look over here on this graph on the left, The number of character differences or tick marks is on the x-axis, and the time since the common ancestor is on the y-axis. So if we compare species B and A, this says there are 4 mutational differences between them. So compare them. They have this common ancestor. We see 1, 2, 3, 4 mutational differences between A and B right there. All right. Now compare species C and B and C to A. We should see 8 differences. So C, B, and A all share this common ancestor. We have 1, 2, 3, 4, 5, 6, 7, 8. 8 differences between, I'm sorry, do it again. 1, 2, 3, 4, 5, 6, 7, 8 differences between C and B. We have 4 here, 5, 6, 7, 8 between C and A. So they should have shared a common ancestor further back in time because it takes more time for 8 mutations to develop than it does for 4 mutations to develop. Now, let's compare species D to species A and B. So they all shared a common ancestor here. We have 1, 2, 3, 4, 5, 6 differences there, 7, 8, 9, 10, 11, 12, 11, 12. 12 differences. So they should have shared a common ancestor even further back in time because it's going to take even more time for 12 mutations to accumulate rather than 8 or 4. And so this can give us relative times of divergence, okay? Now, if we know the actual rate at which that mutation occurs in that gene, then we can back calculate. If it takes 1 million years for it to occur, and you got 4 differences between A and B, that suggests 4 million years of time since they diverged from a common ancestor. All right, that's the idea with a molecular clock. So let's look at some examples of this. First of all, sequences of some genes mutate at a relatively constant rate, and those are useful for molecular clock applications, but there are other genes where the mutation rate is highly variable, and so you simply can't use those genes for that sort of process or analysis. Let's look at 2 genes that are known to mutate at a relatively clock-like fashion in Hawaiian honeycreepers and in Drosophila. Drosophila is a fruit fly. Okay, cytochrome B in honeycreepers. So that's going to be here on the left, okay? Cytochrome B. So first of all, we know the time since the various Hawaiian islands have diverged from each other. We know how much time it took for Maui to move this far from Hawaii and so on down the line, all right? So if we look at Maui and Hawaii, we see that we have mean sequence divergence or difference of about 0.01% right there, okay? Now, if you look at Oahu and Maui, we know that their time since separation was much more, so about 1.5 million years compared to about 0.5 million years for Maui and Hawaii. So it took longer for Oahu and Maui to separate. They've been separated longer, let's say. And we know that we have mean sequence divergence of about 0.03%, okay? And then if you look at Kauai and Oahu right here, they've been separated for much longer, on the order of 3.7 million years, something like that. And so that's more time for more changes to occur, and we see that they do have more mutational differences between them, about 0.065%, something like that. So more time means more mutational differences, and the fact that all of these points fall in a straight line means that that mutation, or that gene rather, is mutating in a clock-like fashion, a linear, straight line fashion. Look at Drosophila. So here we're looking at the YP1 gene, okay? Same pattern. You see all these comparisons, these sequence differences fall on a straight line that coincides with how long the islands have been separated from each other. The longer they've been separated, the more mutational differences you see between those populations. This population and this population are probably not exchanging migrants, and there's probably not much gene flow going on, so they're evolving along separate lines, and we see that in terms of this line arcing upward, having a positive slope to it. But all the points, the sequence divergence points falling on that straight line indicates that those 2 genes are mutating in a predictable clock-like fashion. which means those genes can be used to estimate time since divergence. Okay, so because there are a number of factors that cause error in molecular clock estimates, they're normally used only for estimating approximate dates of divergence. Okay, clocks are rarely going to be used to determine phylogenetic relationships. So we use physical features to determine the branching patterns, the phylogenetic relationships on evolutionary trees, but then when we can, we use molecular clocks to sort of date those divergences, right? Those separation events on the trees. That's what it's really useful for. And the rates of sequence evolution are often consistent among fairly closely related taxa, but they may also differ a lot between distantly related taxa. So this idea of the molecular clock, it's not perfect. There are some instances where it can be used, but lots of others where it's just not useful at all. Okay, next concept we're going to talk about is that of the gene tree. So we've talked about phylogenetic trees generally as showing, as plotting differences in some character, physical character rather, phenotypic character between different species. We can also do this though with genes, obviously, gene sequences. And when we do that, when we plot differences in gene sequences on evolutionary trees, we're creating what's called a gene tree. It's a phylogenetic tree depicting the pattern of evolution as new versions of a gene evolve through mutation. So it's depicting the evolutionary pattern in the development of new genes from ancestral genes, okay? And so versions of a gene that are produced through base pair substitutions are known as haplotypes. Let me write that down, haplotypes. These are versions of a gene, again, produced through mutation. So an ancestral gene will produce descendant genes, and those descendant genes are haplotypes, okay? All right, so let's take a look. So here we see an example of what's called a haplotype tree or a gene tree for, I believe this is McGillivray's warbler. And so what you see is that we have really two geographic populations of this, one in Mexico and one in the US. And so it appears that there's geographic distinction between them, right? You can imagine the Rio Grande River, for example, running between Texas and Mexico right here, let's say, and maybe you just don't get a lot of migration of individuals from Mexico to the US or vice versa. And so what we see then is a distinctive grouping of Mexican and US genes, or haplotypes rather, from the ancestral gene. And this is actually very common. You very often get these sort of geographic patterns where a major barrier, like a large river or a desert or a mountain range or something like that, or an ocean even, separates different populations of the same species, and they eventually evolve into separate species. That's what happened on the Galapagos as many species migrated out to the Galapagos from the mainland of South America. So this is just an example of a gene tree, or what's called a haplotype network. Again, you see that term haplotype, right? Another example of that is from my own research. What you're seeing here is a figure from a paper that was published based on my research back in, I think, about 2012. I worked with a species of cavefish in Indiana and Kentucky, and one of my hypotheses was, during my dissertation, was that the Ohio River, which separates Indiana from Kentucky, was a migration barrier between the Indiana and Kentucky populations of the cavefish. I suspected they could not migrate across that river because it was such a different environment than the cave streams in which they're found, and the river probably bisected down through the caves and separated the Indiana and Kentucky populations from each other. So we took DNA sequences, both mitochondrial and nuclear DNA, to look at this. And this is a haplotype network, a gene tree, showing that very thing. So first of all, what you see is that the black are northern populations, Indiana populations. The white is southern or Kentucky populations. And what you're seeing here is haplotype networks for 5 different genes. The ND2 gene, the S7 gene, the Rho gene, the RAG1 gene, and the TBR gene. So look at the ND2 gene first. So we see that we have an ancestral gene, presumably, that most individuals have, But some individuals in different caves in Indiana show different versions of that gene, different haplotypes, 1 through 7 here, for example. Then compare that gene to the same gene you see in Kentucky, and we see haplotypes 8, 9, 10, and 11. So completely different versions of the gene from what you see in Indiana. And we have 27 mutational steps between them. So we can back calculate how long it must have taken for that 27 gene mutational differences to develop between the gene in Indiana and in Kentucky. And it turns out that date coincides with when geologists tell us the Ohio River formed, about 1 million years ago. So this is a rapidly evolving gene, a mitochondrial gene. That's why you have so many mutational steps between the Indiana and Kentucky populations. But now look at the S7 gene. In Kentucky, you only have basically this one version of it. In Indiana, you have 2 versions of it. So this is a gene that's evolving very slowly. But again, a distinct collection of haplotypes for that gene in Indiana and a different distinct set in Kentucky. Same thing for RAG1. Same thing for TBR. Same thing for Rho gene. So all these genes are telling us the same thing. The Ohio River is a geographic barrier, and the haplotype network bears that out. All right. Now, what about some problems in reconstructing phylogenies? Well, there are about 4 main problems we're going to talk about here. First of all, delineating characters is not easy, right? Analyzing anatomical characters can be subjective. Are 2 features really different characters? There can be a lot of subjectivity in terms of, objectively or evaluating the physical features of an organism. One person might say they're different, one person might say they're not, and there are grades of change as well. Are multiple changes in DNA base pairs really independent changes, or do they evolve together to preserve a function? That's another question. So there can be a lot of subjectivity in terms of analyzing these characters. Second thing, homoplasy, which we talked about before, convergent evolution, reversal, that kind of thing. This occurs often, and homoplasious characters have to be recognized as such because they can suggest relatedness where there is none. They can also obscure relatedness and tell us that 2 species are more distantly related than they really are. Think about an A base mutating to a C and then back to an A again, right? And third, evolution can erase evidence of earlier evolutionary history. As I said, a particular base position in a gene may change multiple times. This is called multiple hits, and that will erase evidence of earlier changes. A modern species may have lost a feature commonly used to diagnose evolutionary relationships as well, so this can cause problems. But let's talk about that problem of multiple hits. So hopefully you remember from earlier biology courses that we have 3 base sequences in RNA called codons. So for example, ACC. An ACC codon will code for a particular amino acid in a protein through the process of gene expression. Well, the thing about codons is that they can evolve, these bases can evolve, and A can go to a U, for example, or a G or whatever, but these 3 bases don't all evolve at the same rate. Because of the way that the genetic code is constructed, it turns out that sequence differences evolve most slowly at second base positions. So second base positions evolve most slowly, third base positions evolve most rapidly, and then first base positions are somewhere in between. So what you're seeing here is the problem of multiple hits. If we look at the pluses, right? Pluses are third base positions. They can evolve very rapidly. And the reason for that is because you can have 4 different codons that all code for the same amino acid. That's the redundancy in the genetic code, but they all have different third base positions. So you can have an ACC mutate to an ACG, and those might code for the same amino acid. So selection doesn't eliminate that mutation. So what you get then is third base positions evolving very rapidly. Here we have sequence divergence, right? So third base positions, the pluses, evolve very rapidly, but eventually they level off. The sequence divergence between two different sequences will level off because eventually you start getting all these changes and it just all gets washed out. And the changes, at some point, you lose all the information that you can use to compare those two sequences to each other. So that means that rapidly evolving sequences are great for analyzing taxa that diverged recently. Right here we have time, so maybe tens of thousands of years. You can, for analyzing that, you can use a rapidly evolving sequence. But if you want to look at sequences or species that you think evolved over much longer periods of time, you might want to use first base or second base positions. You might want to use, in other words, sequences that evolve much more slowly because it takes much longer for them to reach that saturation point where you lose all the information and can no longer tell anything about them through comparison of their DNA sequences. I know this probably seems a little bit confusing, but we're going to talk about the problem of multiple hits and the different rates of gene sequence evolution a little bit later on. But this is a problem with reconstructing evolutionary trees. Fourth problem is that low levels of interbreeding between closely related species can blur distinctions between those species. You can have closely related species that can hybridize. They can interbreed between one another. And so how do you deal with these problems? Well, this is why you'd want to use multiple characters or multiple sequences to try and get a better handle on what's really going on. Again, more lines of evidence suggesting the same thing presents a stronger argument. So, for example, here we have a mammal phylogeny produced by two research teams. Okay, one compared 1,700 sequences across all these different species. So for the African element, they looked at 1,700 different genes. They looked at the same 1,700 for the armadillo and the same 1,700 for the cow and dog and so on. And this is the gene tree that they got. A different research group looked at 2,000 gene sequences, again from these same species, and they got exactly the same gene tree. Now, had they just compared one gene sequence for all these species and the other group a different gene sequence, a different sequence for a different gene, they would probably find differences in their tree. But because they used so much information across so many different genes, the cumulative result was that they got exactly the same gene tree. So, using multiple sequences here. If you're doing this based on phenotypic characters, like, digit number or scale type or coloration or whatever, then you want to use multiple characters. And you put them all together, they should say the same thing. Okay, that's one of the ways you get around one of these problems of, for example, interbreeding between closely related species, blurring these distinctions. You can use multiple characters or multiple DNA sequences to get at the true story of what's going on. All right, but we also have another problem, which is called horizontal gene transfer, along with hybridization. Okay. Hybridization between ancestral species, in other words, those that diverge from the same common ancestor, can produce new species, and this is called reticulate evolution. Here you see an example of a hybridization event. So if we're looking at the relationship between these four species, right, and we're doing it based on gene 1 and then also on gene 2, we might get a difference. Notice how gene 2 suggests that species A and B are more closely related to each other, whereas gene 1 says no, B and C are more closely related. They share a more recent common ancestor. Well, if you see this kind of pattern, when you're comparing different genes across the same group of species, it could indicate that there might have been a hybridization event between species A and C at some point. So species B might be a hybrid species produced through what's called reticulate evolution. Okay, let me just write that down. So reticulate evolution is called reticulate because it produces sort of a net-like pattern on the evolutionary tree like you see right there. But again, this is just another example of some of these problems that we have to sort of deal with. Another problem is called horizontal gene transfer. So horizontal gene transfer. This is when a gene or multiple genes from one species can be transferred or introduced into the genome of another species. And very often this occurs as viruses, for example, jump from one species to another species. So a virus going from species A to species B can actually bring genes from species A into species B. And those genes from species A get incorporated into the genome of species B. So obviously, if you're using DNA sequences to infer phylogenetic relationships, that can result in spurious or erroneous conclusions. And so, for example, when you're looking at these two gene trees, we have one for the Old World monkeys, the Cercopithecidae, easy for me to say, and the Old World cats, the Felidae. There's good evidence that horizontal transfer of a viro gene from the ancestor of the Old World monkeys, as you see here, to the ancestor of the Old World cats probably occurred. Because when you look at a certain gene to construct this evolutionary tree for the cats, it actually suggests that these cats here, shown in blue, are more closely related to monkeys than they are to these other cats. And how could that be? The only way that that could be is because we probably at some point in the past had a virus that transferred a primate gene, a monkey gene, into the lineage of this group of cats, into their ancestor. And so if you're just looking at that gene, it might suggest that certain cats are closer related to primates than they are to other cats, which of course makes no sense. So these are the kinds of things that we have to look out for. These are the kinds of pitfalls that can cause problems for reconstructing evolutionary trees. Okay, that is it for